Hello children, welcome back to the online classes. Today we are going to start about microbes in industrial products. Before going to that, I would like to ask you about some questions which were discussed in the last class. You should answer those questions in comment box. Please answer those questions and then send me answer in the comment box, okay? Write it down. First question. Name the bacteria which is present in curd. Name the bacteria which is present in curd. And then second one, by which bacteria the fermentation of idli and dosa takes place. By which bacteria the fermentation of idli and dosa takes place. And the third question is, large holes on Swiss cheese is due to release of which gas? Large holes on Swiss cheese is due to release of which gas, and which bacteria is responsible for that? And which bacteria is responsible for that? These three questions are regarding the last lecture. Okay, so please go for it and then answer the questions in a comment box. Okay, let us start with an interesting and then very curious topic of this chapter, that is microbes in industrial products. Okay, in the last class we learned about how actually the microbes are helpful in our day-to-day life, how actually they are helpful in domesticated life of a human being. Now we will just go in a larger scale, means industrial level we are going to discuss about here. Okay, microbes in industrial level or industrial products in the sense, you should think in a bigger manner. See children, yesterday, means in the last class, what I said, preparing of curd or else preparing of idli, dosa, bread, these all are the food contents or else the domesticated foods which we are going to use in a small quantity. So for preparation of the curd, we will be preparing a small vessel like that all. Okay, but for the industrial level, what happens, the whatever the vessel will be there, that will be in a bigger manner, because for the industrial productivity there will be need of a large scale of the vessels and all. Okay, before going to that, how actually these microbes are going to be helpful in an industrial level, let us see. Okay, in industry, microbes are used to synthesize a number of products valuable to the human beings. What children? In industry, microbes are used to synthesize a number of products valuable to humans. For example, I can give you beverages and antibiotics. Okay children, beverages and antibiotics are the two industrial products which are given to your service. Rather than this, microbes are used in many of the industries. Beverages, in the sense, the industry which is made for the production of alcoholic drinks, those all industries comes under the beverages. And then when we are talking about antibiotics, means here in the antibiotics means pharmaceutical industry, that is medicines, the preparation of the medicines. The one of the most popular and then always in a what will say, always the pharmaceutical industry will be in a need, because a human being will be getting always a disease will be there, and then they will be in need of medicines. So that this industry is the very valuable industry, and this industry is mainly depend on the microbes for the production of the antibiotics. Okay children, later on we will be discussing about both of this. Okay, first, before going this, first we will be going we are going to learn about what actually the fermenters, and then how actually they are going to be used up, that is in specification of beverages. Okay, under the beverages we are going to learn, later on we will learn about antibiotics. For the production of beverages, like any of the alcoholic drinks, there will be need of a large scale of the vessel. That large scale of vessel is called as fermenter or else bioreactor. Means what children? For the production of an industrial scale, for the production of an industrial scale, it requires what? It requires growing of microbes, in very large scale, or else very large vessels they are needed. That vessels are called fermenter. That vessels are called what children? Fermenter, or one more name is that is bioreactor. Why I am going to tell you about this in the sense, see, for the preparation of the curd there will be need of a small vessel. Okay, in a small vessel only microbes can be grown, but as we are thinking in a industrial level, we are going to think there will be need of a large vessels. That large vessel is called what children? Fermenter. Fermenter will be a area where the microbes are going to be grown for the production of the alcohol. Okay, and then this fermenter, while going to the process of the preparation of the beverage, this fermenter will be of very hot. Why children? Because all of you know fermentation process. When we talk about the fermentation process, the end product will be ethanol or else ethyl alcohol, CO2, and then energy. You learnt in the first year only. What actually fermentation process or else fermentation children? Fermentation, when from the raw material you will be getting glucose, isn't it? Glucose. When the microbes are going to be act on this glucose, what they will be give end product as an anaerobic respiration. In a respiration, first year respiration chapter, you people learnt about this. Okay, glucose will be converted into ethyl alcohol, and then product will be CO2 plus energy is released, isn't it? Due to the release of this energy in a fermenter, while doing the process, it will be hot. Okay children, and then product will be, you will be getting ethyl alcohol. Okay, now let us see how actually they are going to be prepared the beverages. First they will take a raw material children. Okay, first they will take raw material as a source of what? As a source of glucose, like grapes, some of the fruit juice, some of the cereals they are going to take. The next, when they are taking a raw material, they will be, when they are taking raw material, here we are getting what? Maltose or sucrose or glucose or fructose, etc. Okay, etc. These are the fermented sugar. Okay, these all are what children? Fermentation sugar. From the raw material we are getting fermentation sugar. What are those fermentation sugars? That will be depend on the source of glucose, which one you are going to use, like fruit juice or else cereals or else barley, like that. Which one you are going to use? No, you will be getting any one of this. product. Okay, maltose, sugar, sucrose, fructose or anything. Then for this they are going to add yeast. Which yeast, children's? Same yeast which we are already used in the bread making. Okay, that bread making yeast is called what? Yes, that's good. The yeast which is used for the making of the bread, those are called baker's yeast. If the yeast is going to use in the use of beverages, then it is called brewer's yeast. Keep in mind, children. ಬ್ರೆಡ್ ಪ್ರಿಪೇರ್ ಮಾಡೋದು ಸಲುವಾಗಿ ಏನಾದರೂ ಈಸ್ಟ್ ಯೂಸ್ ಮಾಡಿದ್ರೆ ಬೇಕರ್ಸ್ ಈಸ್ಟ್ ಅಂತ ಕರೀತಾರೆ. ಇಫ್ the yeast is going to be used for the preparation of beverages, then it is called brewer's yeast. Is that clear, everybody? Yes. After using of the yeast, there will be process of fermentation. Children. Again, after the fermentation, you will be getting a broth. That broth, again it will be undergo for the process called non-distillation process and distillation process. When the raw material is gone with this all, no, you will be finally get your beverages, that is beer, wine, and then after distillation you will be getting whiskey, brandy and rum. Okay, why this specification I am going to giving you? Based on the process after fermentation, there will be the production of these kind of the beverages like beer, wine, whiskey, brandy and then rum. You people already know about these all things. See, children, once up to this, from the raw material up to the fermentation, everything will be same. Preparation of will be same. Like we took raw material, we left it for the fermentation by using of the yeast. Then next what happens, whatever the extract we will be getting, no, that extract, if that broth, whatever the broth will be there, no, if that broth is pasteurized and then bottled and sold, means that is called non-distillation. After fermentation, pasteurized, bottled, then it is called what, children? Non-distillation. Here what happens, there will be less alcohol content. Okay, after fermentation the broth will be pasteurized and then bottled and then sold out. That is called non-distillation. Example, beer and then wine are non-distillated, so that in the beer and then wine there will be less alcohol. When we talk about distillation, after fermentation what happens, in distillation process they are going to be used for the distillation, aging and then malting process. Like this it will be, later on I will be explaining about that. Distillation process means they are going to add flavors, they are going to add alcohol content, more alcohol, artificial alcohol content, so that the whiskey, brandy and then rum will be having high alcoholic content, children. Okay, this is what about actually the process of the beverage industry will be there. This is only the main basic process I am going to explain you. Where they will be taking raw material, then next they will be keeping the raw material, whatever the raw materials broth will be there, no, that will be contain glucose. That glucose will be undergo with the presence of microbe. Which microbe? The brewer's yeast. The same yeast which is used for the bread preparation, but there we are calling it as a baker's yeast, but here we are calling it as a brewer's yeast. Keep in mind. And then next, after fermentation what happens, if the production of beer is going on, then what happens, it will be for non-distillation. If the production of whiskey is going on, it will be for what, children? Distillation process. It will be by the distillation process. Okay, I would like to tell you how actually in simple manner I will be telling you how actually the fermenter will be there. See, in your NCERT they gave how actually the fermenters will be there. These are the large vessels of the fermenter, children. In these large vessels there will be the production of alcohol, like 20 lakhs liters of alcohol production will be there in these all vessels. This is a plant, fermenter plant, means overall process where actually vessel will be there, outlet will be there, inlet will be there, everything will be there. Okay, for the cleaning and then for the adding of the whatever the raw material is there, for taking of the alcohol after fermentation, everything will be fixed up, and then these all are the vessels of the fermenter where actually they are going to be fermented for the beverage industries. Okay, this is the fermentation plant. I hope everybody is clear about this. Okay, I would like to tell you about one thing that is other than NCERT. Okay, in Greece, I think everybody know about Greece, isn't it? In Greece, one of the first alcohol drink or beverage to gain popularity was called what, children? Made. What called made, and it is fermented. It is a fermented drink. It is fermented drink made from honey and water. Okay, honey and then water. In Greece, first alcoholic beverage which was popularized was mead. The name of that beverage drink is mead, and it is fermented by what raw material? Which raw material, children? Honey and then water. Okay, nowadays whatever we are going to having, those all beverages are recent beverages, but about the beverages you are going to be here in very years ago, like historically also you can see, like in our ancients they are going to use many of the drinks like shindi, madurapan. They are going to use like Indra, the god Indra is also very lovable to drink madira pan, like that. Means these beverages are not only a recent discovery, these beverages are very old discoveries. Okay, children? Yes, that's good. Microbes, that is microbes, especially which one? The yeast have been used from time immemorial for the production of beverages like beer, whiskey, wine, brandy and rum. Means what, children? This is a statement, pure statement from NCERT. Microbes, especially yeast, the scientific name Saccharomyces cerevisiae. Yes, of course, here also same yeast we are going to use, which is used in the bread making. Okay, the microbes, especially yeast, have been used from time immemorial. Time immemorial in the sense the very years ago, very eras ago, the historical ago. Like example I gave already, the Indra, the goddess Indra is also going to take a drink which is called madira pan, like that. Sindhi, these all are the very ancient drinks. These are comes under the beverages only. For the production of what, children's? Beverages like beer, whiskey, wine, brandy and then rum. Means what? These inventions are not this era. Okay, the beverage industry, the beverage production was very old production. Some will think that in what you will say, in Egypt, everybody of you know Egypt history, isn't it? In history of Egypt, if you can know, there what happens, kings used to give beverages to the workers as a salary. Okay children, kings used to give beverages to their workers as a salary, and those workers are going to give those beverages to the shopkeepers for to get a rice, whatever the needs of the house needs are there. Means they are going to give beers, and then they are getting rice and those all things. This is believance is there. Okay, in Egypt history, if you are going to see, the workers were given salary in the form of what? Beverages. Okay, now next, for the purpose of preparation of the beverages, which yeast is going to use, children? Yeast is going to use, that yeast name is what? Saccharomyces cerevisiae. Don't forget the name of this fungi. Yes, yeast is a fungi, children. From the fungi we are going to use, we are going to ferment many of the beverages. Okay, yeast name is Saccharomyces cerevisiae, which is commonly called as what, children? Commonly called as brewer's yeast. If this same yeast is going to use in the preparation of bread, means what it is called? Baker's yeast. That's good. If the yeast is going to be used in the preparation of beverages, then it is called what? Brewer's yeast. Is used for the fermenting malted cereals. It is used for the fermentation of what, children? Cereals, fruit juice, for the production of what? Ethanol. For the production of what, children? Ethanol. Before only I said about this, isn't it? Ethanol, C2H5OH. This is our alcoholic drink which we will be getting. Okay, now in your NCERT they gave one question, that is the metabolic reaction which results in the production of ethanol by yeast. Which is that metabolic reaction? Yes, everybody. Okay, let me tell you. Okay, the question was asked in the NCERT is the metabolic reaction which results in the production of ethanol by yeast is called what? Which reaction? Okay, now which reaction, children? Answer, yeah, very good, that is fermentation. By the process of fermentation only you will be getting a production which is called ethanol, by use of which microbe? Yeast. Once again I am going to give you the reaction. Glucose, which comes from the source of the raw material, which will be converted by a process called glycolysis. Glycolysis will be common for all the kind of the respiration. It may be aerobic or else it may be anaerobic. Glucose will be first, it will be converted into pyruvic acid or else pyruvate. I hope everybody is remembering. Then next, this pyruvic acid is converted in the fermentation process acetaldehyde. Then next, later on, this acetaldehyde is fermented as a ethanol. So our product is formed. During the formation of the from the pyruvic acid, formation of the acetaldehyde will be giving you CO2. Okay, this is one product and this is one product. This is actually how the fermentation process is going on. Fermentation is a two-step process. First one is this and second one is this. This one, conversion of glucose into pyruvic acid, is called what, children? Glycolysis. Okay. Under the fermentation process, when the glycolysis product will be there, pyruvic acid, if it is undergoing aerobic respiration, that reaction will be there. If it is undergoing anaerobic reaction, this reaction will be there. Okay, that's what, once just glance a first year syllabus, that is the respiration in higher plants, in that you are able to see. Okay, pyruvic acid is converted into acetaldehyde is later on converted into ethanol. This ethanol only is used for the preparation of the beverages. Okay, now next, now let us move. See, depend on the type of raw material used for the fermentation. Depend on what, children? Type of raw material which we are going to use. Depending on the type of raw material used for fermentation and the type of process and the type of processing with or without distillation. Already I discussed about this. With or without distillation, different type of alcoholic drinks are obtained. Okay, obtained. Now you should only say, isn't it? Wine and then beer are what, children? Wine and beer are distillated or non-distillated? Yes, very good. They are the produced by without distillation process. Without distillation in the sense what, children? They will be taking raw material, they are going to mixing it with the yeast. Fermentation will be going on. After fermentation, whatever the broth will be there, that broth will be pasteurized and then it is directly bottled. Okay, now means without distillation there will be no any flavor, adding of the flavor, nothing will be there. So here what happens, the alcohol concentration will be what, children? Low alcohol concentration. Okay, next, when we talk about whiskey and all, whiskey, brandy and rum are produced by what? Yes, very good. Are produced by distillation process. Distillation of what? Fermented broth. Fermented broth in the sense, after the fermentation, whatever the liquid will be gained, no, that is nothing but the fermented broth. Okay, now because of this, here the alcohol concentration will be what, children? High alcohol concentration will be there. I hope everybody is understanding here. See, in the preparation of the beer and then wine and then beer without distillation means low alcohol concentration will be. I will be explaining you in detail. Okay, let me take as one example. Okay, beer. Okay, beer is used. When the preparation of beer is going on in the industry, what happens? It will be taken by a preparation of malt, barley. Here raw material used is barley. Okay, for the preparation of what? Beer I am going to talk. For the barley, when the raw material is took, the raw material is having already glucose. Okay, this one will be having the glucose or any source of this sugar it will be having. Barley, then it is added to the barley, it is added the yeast, or else one more name is, children. Yes, very good. Brewer's yeast. Okay, that is Saccharomyces cerevisiae. After this, after the adding of the yeast to the barley, what happens? Fermentation will be there. They will be lefting it for the fermentation. They will be leaving it. Okay, after fermentation, what happens? Now tell me. Now we are producing what? Beer. Whether they are going for the distillation process? No, childrens, because beer comes under without distillation. So after fermentation, whatever the broth will be there, no, that broth is pasteurized, and then next it is bottled and sold out. Okay, yes, that's very good. If I talk about whiskey, brandy, rum and all, what happens? Same will be there. The raw material for the whiskey, it will be of cereals. Many of the cereals are used to for the preparation of the whiskey. They are going to cereal, and then for the cereal yeast is added. Then next go for fermentation, and formed fermentation broth will be what, childrens? It is distillized in the sense the flavor is added, the more content of alcohol is added. Okay, then next it is leave for the aging in the sense, this is for beer. One more example I will be giving you for your clearance. Whiskey. For the preparation of whiskey, the raw material took is cereals. They will be taking cereals. For the cereals, we are going to add yeast. After fermentation, what happens, children? Fermentation, the fermentation broth will be occurs. This fermentation broth is go for distillation process. After distillation, there will be a process which is called aging. Aging in the sense, in a large tankers of wood. Which wood? Oak wood. In the large tankers of oak wood. This is oak wood tanker. Okay. In this aging process, not all the distillated drinks are made by this only. Few are made by like this. Which drink which is made by aging process, it will be very costly. Okay, now here aging in the sense, in this oak wood, what happens? The broth is added, and then it is left for 20 years, 10 years, 15 years, 20 years, 25 years, 50 years, like that. Okay, now how much year it is going to be old, no, that much fermentation will occur, that much taste will, that much flavor will be released from that microbes, because of whenever the age is going on more, that much of rate of that drink or else that beverage is going to be more. Okay, this is one of the example, children. Okay, now some of the I am going to give you some of the alcoholic beverages with substrate and concentration of alcohol. How much concentration of alcohol will be there, and then from which substrate actually that drink or else that beverage is going to be obtained. This here we go. I hope everybody is getting clear picture, and then please note it down. Please note it down. Okay, here some of the list of alcoholic beverages are there, which with the substrate used and then concentration of alcohol, both the table is there. Okay, this is the table which is showing alcohol name, substrate used for the preparation of that alcohol, and then concentration of alcohol present in that. First one I talked about this, that is beer. For the beer, barley malt is used and the alcoholic concentration will be 3 to 6%. The very lowest alcohol concentration. Why? Because beer is prepared by the non-distillation or else without distillation process. That's what the concentration of alcohol is very less. Next, when we come about the wine, that is fruit juice. Wine is prepared from the raw material which is called fruit juice, any of the fruit juice like grapes and all, and then here the alcohol concentration will be 9 to 12%. These two, beer and then wine, are prepared what children's? Without distillation process. Without distillation process. Because of that, there will be low concentration of alcohol. When we talk about the whiskey is used to prepare. Raw material used is fermented cereals. Cereals were fermented here. All kind of cereals like rice and all, they are fermented, and then here alcohol concentration will be 50%, children. The next, brandy. Okay, brandy is for the preparation of brandy, substrate used is fruit juice, and then alcohol concentration will be 60 to 70%. For the rum, molasses, and then alcohol concentration is 40%. Molasses in the sense, molasses is a process, children, where the left out sugar cane, okay, whatever the sugar cane juice will be taken out and then left out sugar cane plant will be there, no, that will be boiled in a high temperature. That boiled high temperature will be giving you a liquid manner contained output will be there. That output is, that process, overall process is called what? Molasses. By that process you are going to get a rum, and then in the rum there will be percentage of 40% of alcohol. Tequila is prepared by use of the substrate which is again a fruit again, and then in tequila there will be alcohol concentration of 35 to 55%. Vodka, a very famous beverage drink, I think so. Vodka is prepared by the substrate used here, it is potato, and then it will be having 60 to 80% of the alcohol. Okay, now children. Then Scotch. Scotch is prepared or is produced by the use of substrate grapes, and then in the Scotch there will be concentration of alcohol, only 63% of alcohol will be there. Okay, now these all, see, as you see, alcohol concentration is more. Then tell me children, these are distillated or non-distillated? Yes, these are the distillated beverages. So high concentration of what, children? Alcohol will be there. That's very good. Okay, this is all about the beverage industry. In the beverage industry, under the beverage industry, we learnt about how actually beer, wine, whiskey, brandy, rum, tequila, vodka, scotch, these all, many of the brands are there, many of the things are there which are going to be produced in a large scale. In a large scale, in the sense, there will be production of 2 lakhs together liters of the beverages per fermentation, like that it will be. Okay, now this is all about the beverages. And then process, already I explained you how they will be preparing. First they will be preparing stage will be there. In that raw material they will be taking. Second one, masting in a sense mixing, whatever the raw material will be there, no, that will be mixed with the microbe. Then next it is undergone for the fermenting. Then next distillation. If there is a production of the distillated alcohols, then it will undergo for the distillation process. Otherwise, here only it will be stopped for the non-distillated process. Okay, after fermentation, directly they will go for pasteurization and bottling and bottled and sealed. This is for whom? This is for non-distillation. This is for without distillation. Without distillation. And the next process will be for with distillation. Like after fermentation, distillation process will be there, means adding of the flavor, adding of some of the alcoholic concentration and all. Next there will be aging. Already I said, in a large tankers of oak tree, oak tree tankers will be there. In that oak tree tankers, what happens, the fermented broth will be added and then it is left for many of the years. Okay, for example, I can say you one of the very famous brand of John Walker, the Walker's grandfather, Johnny Walker's grandfather, where in before 50 years ago he kept the whiskey fermented broth in a oak tanker. Now the John Walker is selling those all the whiskey brands. Okay students, I don't know much about this, but historically in Google if you going to search, you will be getting much more idea about this. That is called aging. Then next, after aging they will be blending it, then next bottled, then next it is going to be sold out. Okay, this is all about alcoholic beverages, childrens. Okay, I hope everybody is clearly understood about this. If anybody is having any doubts, clear doubts, please ask in comment box. Okay, next about the next, see children, we learnt about beverages. Next comes what? Yes, the interesting and then very essential knowledge one should have, that is about antibiotics. Okay children, antibiotics. Each and every organism or human being is depend on antibiotics one or the other way, so that gaining knowledge about the antibiotics is very essential. Okay, let us see what actually antibiotics are. Antibiotics, they are chemical substance. They are what children? They are chemical substance which are produced by some microbes, and kill or retarded the growth of other microbes which are disease causing, which are what children? Disease causing microbes. Already in the first class only I said you about these microbes. All the microbes are not disease causing and all the microbes are not useful also. Some of the microbes are disease causing and then some of the microbes are what? Helpful. Here in the antibiotic, the main theory is that one only. The antibiotic is a chemical substance which is produced by some microbes. For what children? To kill the growth of other microbe which is going to cause a disease. If I say you like this, you won't be understand. So I would like to tell you a story with an example. Okay now we are there, that is we the human beings. Okay, next there is a microbe which is, I would like to give you a name as bacteria A, and one more microbe is there, bacteria B. Okay, this bacteria is disease causing and this bacteria is useful bacteria. Okay, this bacteria is what children? Disease causing bacteria, and this is, yes, this is useful bacteria. I can say useful bacteria or else microbe. Now what happens? They will be interacting with each other. When they are exposed to each other, then what happens? Some of the chemicals which are released by useful bacteria will be released against the disease causing bacteria. That chemical itself is called antibiotic. Okay, that this is a chemical substance which is used by the useful bacteria. This is also life only, this is also life only, this is also organism, this is also organism, and this is also organism only. But here what actually happens, students, dushman ka dushman dost. For this bacteria, who is the enemy? This bacteria is enemy. If this bacteria is going to cause disease for the human being, if this bacteria is going to cause the any of the disease to the human being, for human being also it will become what? Dushman, that is enemy. It is also become enemy. For the killing of this enemy, what you should use? What we should use? The chemical which is produced by our friend. Okay, is that clear? For the human being, this bacteria is enemy now, isn't it? Yes, and then this bacteria is already enemy for any of the other bacteria which is useful for the human beings. So now tell me, which will be antibiotic? Whether the chemical which is produced by this? No, the chemical which is produced by this kind of bacteria is called what? Antibiotic. Okay, na? Why? Because this bacteria, see, I will be explaining you in simpler manner. See, this is bacteria A. Just imagine, okay? Don't laugh. This is a bacteria A and this is bacteria B. Which is our friendly bacteria? This is our friendly bacteria and this is enemy bacteria. These two will be having always fighting together. Whatever the chemical is released by this will be having against chemical which is released by this bacteria, friendly bacteria. Okay, now if this bacteria is going to attack on human beings, if this is going to be attack on human beings, who is going to save? The friend bacteria is going to be saved. Okay, by the release of this bacterial chemical, what happens? This bacteria's effect and growth will be retarded. Okay, that's what it is written here in NCERT. It is clearly there. Okay, for the saving or else for the kill of the whatever the chemical, whatever the disease is caused by the disease causing microbe will be retarded by whom, children? By the microbe which is giving you antibiotic chemicals. Okay, that's good. Now I hope everybody is clear about this. Now let me tell you something interestingly, that is here anti in the sense in Greek words, anti in a Greek it means against. Okay, anti means what, children? Means in a Greek anti means against, and bio means everybody you know, that is life. Okay, in according to Greek, anti means against and then bio means what, children? Life. So together it means what? Together it means against the life. Here please don't get confused. Here against the life means against this life, not this life. The antibiotic which is released by this microbe not against the human beings, but it is against to the disease causing bacteria or else disease causing microbes. Okay, now together that can be called as against life. Here against life in the sense in context to, in clearly in NCERT they gave in context of disease causing organism. Disease causing organism. Okay na, disease causing organism. ಈ ಡಿಸೀಸ್ ಕಾಸಿಂಗ್ ಆರ್ಗ್ಯಾನಿಸಮ್ಗೆ ಆಂಟಿಬಯೋಟಿಕ್ ಎಲ್ಲಿಂದ ಬರ್ತದೆ? ಈ ಬ್ಯಾಕ್ಟೀರಿಯಾದಿಂದ ಬರ್ತದೆ. ದಟ್ಸ್ ವಾಟ್ ದಿಸ್ ಕೆಮಿಕಲ್ ಈಸ್ ಕಾಲ್ಡ್ ವಾಟ್? ಆಂಟಿಬಯೋಟಿಕ್. ಓಕೆನಾ? ವೇರ್ ಆರ್ with reference to human being, they are pro-life. They are pro-life, not against. Okay, they are not against. Okay, they are not against. If I ask you, this bacterial chemical which is releasing for the human being, whether it is useful or harmful, useful only. That's what it is, pro-life. If I ask you, this bacteria is releasing whatever the chemical, whether that chemical is useful or harmful for this bacteria, yes, of course it is harmful. Because of that, it is antibiotic or else against the life. For this against, for this pro-life, this antibiotic. Okay, is that clear? Yes, everybody, very good. And here the antibiotics are regarded one of the most significant discovery of this 20th century. Okay, in the 20th century, see children's, antibiotic discovery is a chance discovery. By purposely it is not at all discovered, it is a chance discovery. I am telling you now only. One of the very regardable discovery, significant discovery of 20th century is what, children? The discovery of the antibiotic. And then in the 20th century, in the sense from the year of 1927, it is started actually. Okay, now the work about the antibiotic work is started from the year 1927. But for the starting, after the getting of this antibiotic and then after getting released, it took 20th century. That's what the remarkable, regardable discovery of 20th century is, the antibiotic. Okay, now very interesting thing about antibiotic. The first antibiotic was penicillin, was discovered by Alexander Fleming. Okay, first antibiotic penicillin was discovered by whom, children's? Alexander Fleming. He started in 1927 only, and then after many of the works, on September 20, that is 1928, he showed that about the antibiotic penicillin. I will be telling you overall story about this, how actually it is going to be discovered and all. Okay, now next, this is how actually the Alexander Fleming were born, how actually he discovered. I said to you, this is not it's just a chance discovery. What, children? The discovery of the penicillin itself was a chance discovery. Okay, now how it is a chance discovery? See, when Alexander Fleming was working on, while he working on a bacteria. Which one bacteria? Staphylococcus bacteria. He was working actually on this bacteria. Okay, working in the sense in a lab condition. They are going to take a Petri plate and then they are introducing that bacteria in that Petri plate, and then they are going to isolate which kind of chemical that bacteria is going to be released and all. Okay, now while he working so on this kind of bacteria, he observed that a mold growing in one of his unwashed culture plate. Okay, now unwashed culture plate, where around which around which staphylococci could not grow children. Means what? He found that the chemical which was produced, he found that the chemical was produced by fungi. The chemical which was produced by fungi, which is helpful, which is not at all allowing to grow of this bacteria, and later on that fungi is named it as what, children's? Penicillin. Okay, what actually this thing is given in your NCERT. Now I will be telling you story what actually happened with the Alexander Fleming. See, in a lab he was working in a two working manners. First work was on bacteria. Okay, first work was on bacteria, that is in a Petri plate. He used to grow bacteria which is called what, children's? Staphylococci. Okay, one more, in another manner he was working on a fungus, or else fungus he was working on, but actually he was not knowing that fungus name actually. Okay, that's what he used to call it as a mold. Okay, now whose Alexander Fleming's assistant was there, who called Mary. Alexander Fleming discovered all this in a London, children's. Okay, now in London Alexander Fleming was having a assistant. Her name was Mary. She used to go for the bakery, and then he used to take old breads. Okay, now old breads, because in old bread there will be growth of the molds, or else there will be growth of the fungi, and that fungi is used for this experiment. Okay, and then for Staphylococcus, other bacteria is going. Actually, this is another work, and this is another work. Okay, when these both the works, this is the growth of the fungi on the Petri plate when they are going to be worked out. Next, what happens after the work happened? What happens? They kept it for wash. While doing so, what happens? Some of the molds, some of the fungi, what happens? It is exposed to the Staphylococcus. Okay, some of the molds by chance, not by purposely, by chancely, these molds were exposed to the Staphylococcus containing Petri dish. In this Petri dish, what happens when Alexander Fleming came in next day, and then he saw this Petri plate, he saw how come this mold is grow on this, and then this mold is nothing but what, children's? Fungi. Okay, now around this fungi, whatever the fungi is releasing the chemical, around this fungi there was no growth of this Staphylococcus. It means what? This fungi is retarding the growth of the Staphylococcus. After this he repeated this experiment by purposely many times, and then he concluded that yes, this fungi is having a chemical which is releasing against the Staphylococcus growth. So that in that year the penicillin was discovered. For after this, its full potential as an effective antibiotic, this is only the discovery. Okay, now but potentiality means after this repetitive experiment, the Alexander Fleming took help of other two scientists, that is Ernst Chain and Howard Florey. These three, Alexander Fleming, Ernst Chain, Howard Florey, they were discovered effective antibiotic. Okay, now they were discovered effective antibiotic, and then I will be telling you means after this discovery what happens, children. See, before this discovery what happens, actually I will be telling you. In World War First, what happens whenever any of the soldier is going to be injured. For example, if any of the soldier is injured here, okay, by bullet or something, what they used to do, because the injured part will be exposed to the staphylococcus, because staphylococcus will be present in the soil, air, everywhere. Okay, when it is affected, no, the soldiers, the palm or else hand will be cutted overall, because that will be spread all over the body. To stop that spread, what happens, they used to cut that organ part, or else they used to cut that area where actually infection is going on. This is in the World War First. After, in between that, when this discovery is going to be happen, luckily what happens, children, in the World War II, in World War II, the American soldiers were treated. American soldiers were treated their wound by penicillin. Okay, na children? So that, and the 20th century, this was a very, very, very gratitudable discovery, it is said. Okay, this is about how actually the the discovered penicillin was used in the World War II. Before that, in the World War I, due to the wounding, only due to the effect of the staphylococcus only, there were many of the soldiers are there which they lost many of their organs. Okay, now because of this discovery, the Fleming and Chain and Florey were awarded the Nobel Prize. In 1945. In 1945, they awarded the Nobel Prize. Okay. After penicillin, actually this penicillin was discovered, means this penicillin is a fungi, children, already I said. After the discovery of penicillin, many antibiotics were there also produced from other microbes like bacteria and then Actinomycet. Not only from the fungi, many of the antibiotics were produced from bacteria and Actinomycetes. This Actinomycetes is nothing but a soil bacteria, children. Okay, now this what just now I explained. No, this one penicillin is nothing but it is a fungi, children. Okay, after this discovery, not only from the fungi, other than that fungi, bacteria and then Actinomycetes also, there were the production of the antibiotics. Here you will be wonder that from the Actinomycetes there are 2100 antibiotics were produced nowadays. 2100 antibiotics were producing from this Actinomycetes. Actually, this Actinomycetes is a soil-borne bacteria. Okay, now in your NCERT they gave one question, that is, can you name some other antibiotics, and find out their source. Okay, means, see, now we discussed about penicillin. Okay, now other than the penicillin, some other antibiotic you should find out. Then next, from where actually that antibiotic source is, that means from which microbe actually it is going to be formed, that one you should be make a list. Okay, make it off. Then next, when you will be searching for this question in Google, if you search, you will be getting the answer. Tomorrow or in the next class, in the next session, if anybody is answered, then I will be declaring their name. If nobody, means I will be answering for this. Okay now children? Yes. Next, antibiotics have greatly improved our capacity to treat deadly diseases. With antibiotics, okay, after the discovery of antibiotics, have greatly improved our capacity to treat deadly diseases, like such as many of the diseases are there, like plague, cough, diphtheria in the sense galugotu, you will say leprosy in the sense kushtarog. One of the era was there, one of the century was there where this kushtaroga was very, very, very widespread. Okay now, due to the antibiotic effect only, nowadays you cannot even able to hear the word of kushtaroga. That everything credit goes to the antibiotic pharmaceutical industry only, children. Okay now, and then we cannot imagine a world without antibiotics. Yes, of course we cannot imagine a world without antibiotics, because for each and every small big diseases there is a need of antibiotic, children. For example, if anybody is injured, if anybody is got fever, if anybody is under a large disease, a big disease, they are in need of what? Antibiotics, because most of the diseases were occurred due to the microbe only, which is affecting on the human beings. To fight against that microbe, we are in need of microbe which is releasing the antibiotic. Okay children? Yes, this is all about today's lecture, and hope everybody is clearly understood. And then if any question, if any doubt is there, please comment on, and then please follow and subscribe.